Thanks, Josh, for that kind introduction. Thank you also for your and Walter Korshetz's leadership of BRAIN before I came on. I wouldn't have come on had you not done such a great job in getting us to where we are right now. And also, of course, thank you for the opportunity to give this presentation to Council. I think it's very exciting. I hope you share in the excitement with me as we go along. So what I wanted to do here was just to give a very brief, and emphasis on brief, overview of where BRAIN is going for this next phase, the so-called BRAIN 2.0 phase. But just to remind us, or to remind me, is this going to work? Here we go. To remind me about what our big picture goals are. And this is really to develop and apply new tools for understanding how neural circuits underlie behaviors, both in the healthy brain and in various model systems, as well as in models of disease and ultimately in human disease. And I believe that one way to help us achieve these goals is to keep our mind on three things. One is we really need to continue leveraging technology to enable new discoveries about neural circuit function. We need to use these discoveries as a foundation for new therapeutics for human brain disorders. And third, and I really want to emphasize this last point, we really need to focus on disseminating and democratizing these technologies, both for basic discovery as well as for clinical applications. So BRAIN was launched in 2014. It was announced in 2013, and it was launched based on a really thoughtful strategic plan that was written and released in 2014. This plan was put together by a working group of the ACD, Dr. Collins' advisory council to the director. They issued this so-called BRAIN 2025 report. The chairs of that committee were Corey Bargman and Bill Newsom. And about 5 years in, I think as with any good organizations, it was decided to take a look at where we stood about 5 years in, take a look at the strategic plan, brush it up, consider the accomplishments that were made and maybe some tweaks that could be made moving forward and what really were the new opportunities. So this resulted in the BRAIN Working Group 2.0 that was appointed by Dr. Collins, a group chaired by Catherine Duloc and John Mansell, and they issued their findings in October of 2019. And I just want to highlight for you a couple of the main takeaway points from that report, because it really does inform how we want to proceed over the next 5+ years. The first was to stay on the productive path. I refer to this as keep on keeping on, keep on keeping on. A lot of things have been happening that have developed some really terrific and powerful tools. They have led to a lot of great discoveries. I think it really behooves us to continue on that productive path. We want to make sure we maintain or even increase an emphasis on making discoveries with these tools. The next major bullet is to remember to continue or place an even greater emphasis on behavior, parts of the brain that aren't the cortex, and also to leverage the power of model organisms where they can be appropriately used either to develop new tools or to reveal new fundamental principles. And then the third bullet is to balance individual investigator-initiated research with team science. Now, as I'll show you for most of today's talk, there's been a great amount of progress made in certain areas that could only have been made by doing large team science. But I think one of the hallmarks of the NIH is the discovery or the discoveries being made by ideas that are formulated by smaller teams and tested through rigorous hypothesis-driven research. And presumably the tools that we are developing can allow for better and more powerful hypotheses to be tested in a much more rigorous way. So we want to balance our efforts that way. And then the final point, and this will be the major topic of today's discussion, was a suggestion that given where BRAIN has come over the last 5-6 years, all the discoveries and developments that have been made, both within BRAIN and outside of BRAIN, now is a great time to ask, can we formulate and stand up large-scale projects that could truly transform the way we perform neuroscience research and ultimately in the way that we treat human brain disorders. So this is what I'm going to focus on for the rest of today. But to put this in context, here's what the outlook for BRAIN looks like from its inception through 2026. Again, started in 2014 with a rather modest NIH budget of $46 million. Right now, we're toward the end of fiscal year 2020, and we're expected to spend around, or invest, I should say, on the order of $500 million, which was the amount that the BRAIN 2025 report recommended as our funding goal. We're about a third of the way through a total investment of $5.2 billion through 2026. Note that we have a base funding plus generous but uneven funding from the 21st Century Cures Act, which results in a very large bump up in our funding in fiscal year 2023, where we're expected to invest on the order of $810 million. So this is our target. We need to build a runway to fiscal year 2023 to really make sure we take best advantage of this increase in funding resources available to us. And this is the basis for our planning for these, what I'm calling the BRAIN 2.0 transformative projects. Okay, so the idea is to build on the solid foundation of discovery and technological innovations that we've seen in the preceding 5 or arguably 10 years. I want to walk you through 3 major projects that they interlink that we're going to be launching. And what I'll do today is I'll just, after summarizing these projects very briefly, I'm just going to give some scientific highlights that show some of the rationale and the foundation and motivation for these 3 large projects. And hopefully we can discuss at the end what the future might look like based on these foundational insights. So the first is the phase 3 of the brain cell census. This is pending concept clearance a bit later today in front of you. And the idea is to build on the success of the BICCN, the Brain Initiative Cell Census Network, which has focused mainly, but not exclusively, on building a comprehensive cell census of the mouse brain. And the idea is in the third phase is to shift emphasis to construct a comprehensive human brain cell atlas. And Dr. Yong Yao, who's been shepherding this project all along to great effect, will be presenting to you this concept for clearance a bit later today. The second major project we are planning to promote is this development of next generation technologies for performing brain microconnectivity analysis. This is basically creating a wiring diagram of the brain, a complete brain, at multiple scales and possibly in multiple species. So the idea is between these 2 projects, we hope to have some ground level truth about the parts of the brain, the components, the cellular components, both neurons and non-neural cells, as well as how these cells connect up with each other. And this should give us a really great foundation for probing not only how the brain works normally, but for probing and identifying disease-relevant circuits. Now, the third one will build on the discoveries made here in the first 2 projects, which is to develop resources for gaining access to cell types in the brain for both monitoring as well as for manipulation in multiple species. So we refer to this as the cell type specific armamentarium, basically a large toolkit to gain cell access into cells, not only in rodents where transgenic technologies are readily available and easily applied, but also in model organisms that are less amenable to transgenic analyses, germline analyses, including non-human primates, and ultimately to develop these tools toward human cells and tissues and eventually into humans themselves. The long-term goal of this project is to lay the foundation for developing new strategies for human brain disorders along the lines of molecular and gene therapies. So again, these first 2 projects will give us some ground truth about what brains look like in terms of their parts, organization, and connectivity. And this third project will give us, and actually enable these projects, but give us tools for interrogating and manipulating circuit function based on cell type specific information. So let me just go through each of these projects one by one and give you a little bit of background of why we think this is important and why we feel that this is the right time to launch these projects. So the first is the brain cell census project, again, which we intend to move into humans over the next 5 years. And the motivation for this project is simply we need a parts list for the brain. How does this remarkable computer function? In order to know how it functions, we need some fundamental knowledge about what comprises this biological computer. The reality is that this has been a problem that's been around with us since the days of Ramon y Cajal over 100 years ago. And we really need to develop a systematic way to identify and classify these cell types in order to understand what their functional properties are. So the idea behind the Brain Initiative Cell Census Project is to build an integrated brain cell atlas based on multiple modalities. Now, many of you have probably heard about the advances being made in single-cell sequencing, which has truly revolutionized our ability to characterize and classify cell types throughout the body, but it doesn't begin and end there. So while much of what we're doing in the Cell Census project right now is relying on molecular signatures as a foundation, mainly single-cell RNA sequencing, single-cell epigenomics, including ATAC-seq, methyl-C sequencing, et cetera, this is really just the beginning of the journey where we're going to use this information to help us get more information about each cell type's anatomy, their connectivity, and ultimately their function within neural circuits. So why do we think, okay, so where are we in this process? The Cell Census project started in 2014. It was one of the first sets of projects funded by the Brain Initiative. I was very fortunate to be among the first projects in the Brain Initiative Cell Census Consortium, or BICCC. Up until about 3 months ago, I was a PI in the next phase, which I'll talk about in a moment. The goal of the BICCC at the time in 2014, which was many lifetimes ago, was to evaluate, validate the various emerging technologies that were just coming online, mainly, but not exclusively, in the single-cell sequencing domain, and to see if they could be scaled in order to obtain a comprehensive atlas of an entire brain, for example, the mouse brain. And this led eventually to the scale-up phase, where we are currently, phase 2, which is the Brain Initiative Cell Census Network at BICCN. And again, I was a PI in this group until about 3 months ago. And the main goal here is to develop a comprehensive mouse brain atlas, but also to begin some pilot studies in human and non-human primates. We're about halfway through here. This is expected to run through the end of 2022. And then phase 3, which Dr. Yao will tell us about a bit later today, will involve ramping up into the human brain. Not to ignore other ongoing work in humans and non-human primates, but here in phase 3, the main emphasis will be into moving into human brain. So we're right about here. Let me just tell you a little bit about the BICCN. So the BICCN is one of the larger, if not the largest, organized consortia within the Brain Initiative. It's a bit of a juggernaut, as this diagram.
Forwarded to an automated voice messaging.
It's a bit of a juggernaut based on this figure that I'm showing here. It's centered around 3 large U19 production grants, as illustrated here, that are focused mainly on generating data and analyzing the data. There are a series of significant but somewhat smaller U101 grants down here, again focused on the mouse, that are focused primarily on developing new techniques and applying these techniques for understanding connectivity based on this integrative analysis. We have a series of other U01s that are focused on both human and non-human primate brain, another series of R01s that are focused on technology development, and all these studies are coordinated through a coordinating center here, whose goal is to both coordinate the activities as well as to ensure that the information coming out of these studies will be deposited in a way that is accessible and usable by the larger scientific community. So this is the BICCN. You can learn a lot more about it by going to this website here. Okay, so what have we done? This is a large effort. You can see just by the number of groups involved, many of these U19 and U01s are multi-investigator, multi-site projects. We decided that we needed to make sure that we could work effectively together, not just from a social point of view, but from the point of view of can we actually come up with a consensus view of what the brain looks like based on not only multiple modalities, but multiple modalities being carried out by multiple sites. So it was decided that one way to test this, this notion was to have us all focus on one region of the mouse brain, which is the primary motor cortex. And what I'll do is I'll just highlight for you 4 studies that were conducted that show that I do believe that this has been remarkably successful. This is 4 out of, I think, over a dozen studies that have recently been deposited in bioRxiv. So I'm only giving you a very high level highlight. So the first was to determine whether we could conduct an integrated analysis based on the data that were being generated from multiple RNA sequencing platforms, as well as epigenomic platforms from multiple sites. These were from the U19s that I mentioned to you, This is just a highlight from a bioRxiv preprint that was deposited from a group led by Honghui Zhang, who's here on council, as well as Aram Mukamel from UCSD. There were 83 authors on this paper. I was one of them, maybe about over a dozen groups involved. And the answer was we actually came up with a very compelling integrated analysis based on both single-cell transcriptomics and epigenomics that, depending on whether you're a lumper or a splitter, leads to a resolution of between 70 and 130 statistically distinguishable groups of cells. Now, in a project led by Xiaowei Zhuang at Harvard, who applied her MERFISH technology for conducting spatial transcriptomics, this allowed us to place the identities of these cells into anatomical position within this laminar cortex, both providing additional information for where these cells are on top of validation for these cell types, but also gave us additional information about their projection patterns. Andreas Tolias at Baylor University, a pioneer in this technique called patch-seq, was able to integrate many of these modalities together by performing patch clamp recording from individual cells and slices. He could actually obtain information about the cell's physiology. By removing the RNA from those cells, he could then sequence it and lay that sequencing information onto this framework provided by the single-cell RNA sequencing provided by the other groups. And then finally, by filling the cells and performing microscopic analysis, you could also obtain information about these cells' morphology. And then in a group led by Ed Lean at the Allen Institute, the study was extended into non-human primates as well as into humans. And the remarkable discovery here is that in spite of this very large difference in size and presumably in terms of computational complexity, there is quite a bit of conservation in cell types between non-human primates and rodents. So it means that the efforts that we've been putting into the mouse brain will actually serve as a good foundation for looking at these larger brains. But it also pointed out some key differences which point the way toward both evolutionary differences as well as differences in development and presumably also differences in terms of function. So again, this is just a very brief overview. I'm only covering a very small number of the studies that are now shown and deposited in bioRxiv and are under review. And hopefully we'll see these papers out within the next year or so. So in summary, the goal of the BICCN is to build a comprehensive brain cell atlas based on multiple modalities, again, based on molecular information to derive, plus morphology to derive data-driven cell taxonomies. This information in turn can be used to develop cell targeting tools that can then be used to map connectivity as well as to interrogate the roles of specific cell types in circuit function. So let's turn our attention now to this connectivity project and the basis of this microconnectivity project that we are moving forward on in the next phase of BRAIN. One of the long-range goals of the BRAIN Initiative and neuroscience in general is to link circuits to behavior. I don't have time to tell you about a lot of the incredible developments that have been made in terms of developing new actuators and sensors of cellular activity. Many of these are genetically based. Some of these are based on other modalities, including nanoscience. There's been the development of many different powerful imaging modalities, both optical and otherwise, to visualize activity within neural circuits, often in awake behaving animals. And again, this is really driving toward the goal of obtaining principles underlying the circuit basis of behavior. And this really sets the stage for why we really do need high resolution maps of connectivity within the brain. Again, it will provide a ground truth for circuit analysis. We need maps that span both the meso and the nanoscale. So in other words, what are the connections between brain regions? We're seeing a lot of really interesting studies showing that complex behaviors engage different regions of the brain to generate the output. But we also need to understand what the local circuitry is as well at the nanometer or synapse scale. It's very important to look at both dynamic as well as static maps. So by this, I mean, we not only need a map of what the connections look like, but we need to understand what the traffic patterns are that represent the information being carried by those maps. And we also need to include in these analyses cell type specificity. This is information that will come from the Cell Census project. So let me just give you a very quick example of where we might be able to go with this and how would it be further empowered by a deeper understanding of connectivity maps. This is a summary of a recent paper from Florian Engert's lab. The Engert lab with others at Harvard are conducting a large U19 grant looking at the connection between sensory input and ultimate motor output in behavior. So here they're using the zebrafish larva as a model system. It's great because you can observe this fish behaving to the extent a fish can behave. You can provide an artificial stimulus. And the real beauty here is that given the size and optical transparency of the larva, one can actually view activity across the entire zebrafish brain. So one can actually observe what's going on activity-wise as this fish receives a stimulus, processes the information through its circuits, and ultimately generates a behavior, which in this case is a swimming behavior. And using this very beautiful experimental paradigm, Engert and colleagues have been able to synthesize this information to propose a circuit motif for decision-making in response to this stimulus, this do I swim right or do I swim left, for example. And they propose this circuit motif, and which can be now subject to further testing and validation. So I think what I'm trying to get at here is that understanding the computations carried out by circuit motifs will be greatly enabled by characterization of the local circuits at the synapse level. So this brings in this landmark paper from Jeff Lichtman, published back in 2015, where Lichtman and colleagues used three-dimensional EM reconstruction to reconstruct a small chunk of mouse cortex, a 1.5 millimeter, 1.5 cubic millimeter chunk of mouse cortex by using serial sectioning and imaging through scanning electron microscopy and a lot of heavy-duty statistical analysis. So this is just a three-dimensional rendering, this pretty movie, and really the challenge will be, given that one can obtain this information at the local level with a lot of effort, how do we scale this up to an entire brain? And for our purposes, given the power of the mouse as an experimental model, we ultimately will want to scale this up to an entire mouse brain, which presents a problem of a physical scale that's 2 to 3 orders of magnitude larger. And in terms of experimentation and computation, it's probably going to scale superlinearly. Okay, so this is what the challenge is going to be. One is how to do this at greater scale, but also how do we link this information from the local circuits to these projections between different regions of the brain? And here I just want to highlight a study that was conducted by Ed Boyden at MIT and Eric Betzig, who's now at UC Berkeley. And this involves a combination of Boyden's expansion microscopy together with Betzig's lattice light sheet microscopy. So the idea is we want to be able to look at connections over large regions of the brain, but we want to be able to zoom in at very high resolution. So what Boyden has done is he's taken this expansion technique where he basically takes a tissue and he expands it in a hydrogel so that features or subanatomical features of the cell are now spread apart in a non-distorted way. So what used to be below the resolution of the light microscope is now resolvable by light microscopy. And then this can be imaged, and if using the lattice light sheet microscope that Betzig has developed, it actually allows resolution on the order of 50 to 100 nanometers, but it allows them to view specimens on the order of millimeters. So this really helps us bridge these scales from meso down to nano. Okay, so this is just kind of a highlight of things that may come in the future based on future developments. So finally, I'd like to talk a bit about this cell type specific armamentarium. In other words, technologies for enabling access to cell types for both experimental as well as for eventual therapeutic purposes. Now, it's going to be important for us to access different cell types in the brain, especially given all the information that's literally pouring out of the cell census projects. How are we going to do this in ways that will be able to access cells in so-called non-genetic organisms? Well, there are ways of accessing cell types in somatic cells using both viral and non-viral vectors. I'm just going to touch on one such vector, the viral vector, the adeno-associated virus vector. Here's a study that was developed by Dr. Viviana Gradnaru at Caltech, who used directed evolution to select for AAV capsids that conferred cell type specific tropism. Now, one of the first set of tools that she made was a virus that was capable of crossing the blood-brain barrier, which experimentally and therapeutically could be a great advance, because one can then target cells in the brain without injecting directly into the brain, but rather into the bloodstream. So access is much easier. They then use this next iteration of the technique, which they call mCreate, to develop further specificity and identify and isolate viral capsids that can either selectively target neurons, astrocytes, or endothelial cells, but also selectively avoid other organs in the body, for example, liver, kidney, and heart. And this is important because one of the issues with some current gene therapies using AAV is toxicity, hepatotoxicity. So this could be of great use, not just experimentally, but in thinking ahead for human therapeutic use. And the beauty of this technique is that it's scalable. One, in theory, can just go after whatever cell types one wants. And that combined with cell type specific promoters or enhancers that are being identified through the Cell Census project, this technology can really give exquisite targeting in both research and therapeutic applications. Now, I mentioned, so here is just some examples of various different cell types that are targeted by one of Gradnaru's selected AAVs. Now, I mentioned early on the importance of democratizing technologies. And currently we're seeing some very exciting news about gene therapies that have been launched and found to be successful in early trials in humans. One of the main problems with these trials is that they're really quite expensive. There's $1 million therapies, and we need to find a way to bring the cost down. And one way to do that is to democratize the technology. And one way to democratize this technology is to view it as a modular toolkit, where there are different shelves in the toolkit. One could involve capsids that are selected for different tropisms. Another shelf in the toolkit can involve promoters or enhancers that further direct specificity to cell types. We have payloads, and together, one can imagine that if one wants to apply a certain therapy or a certain gene editor or a certain type of actuator to a cell type, one can go to this toolkit and use pre-validated and perhaps even pre-approved components, which would make the barrier to entry much, much lower, and hopefully, therefore, the cost lower as well. So one of the limitations of these AAV vectors, and there are other vectors under development as well, but one of the limitations of AAVs is that the payload they can accommodate is quite limited. And many eukaryotic gene promoters are quite large, but one can actually gain cell type specific expression if one can identify enhancer elements that drive expression in specific cell types. And here's just a taste of things to come. This is a study conducted by Ed Lean and colleagues at the Allen Institute. And here they use single cell ATAC-seq to identify regions of open chromatin across brain samples from multiple species. And they use this to identify putative enhancer elements that could be active in both in rodent and non-human primate brains. And they actually showed activity of these enhancers within subsets of inhibitory neurons. And quite excitingly, they can also show activity of these enhancers in human brain slices. So this really paves the way toward thinking about developing these tools for eventual therapies in humans. So let me just finish up by talking a bit about opportunities for interventions in humans. The last part of the title of the Brain 2.0 report was toward cures, and this is really, I think, where we need to be heading. And I would argue that the discovery supported by Brain will lay the foundation for novel interventions in human brain disorders. These technologies are helping us unravel the circuits and computations underlying complex behaviors. This will lead to the identification, I expect, of cells and circuits affected in neuropsychiatric disorders, and presumably identify new targets for therapeutics. We're already seeing the development and application of closed-loop deep brain stimulation devices to address some neuropsychiatric disorders. And eventually, I think we can become more precise about how we intervene through these targeted molecular and gene therapies. And I just want to finish very quickly by summarizing two studies that are currently funded that would be of interest to the National Institute of Mental Health. One is a study being conducted by Helen Mayberg, who's been a pioneer in applying deep brain stimulation, or DBS, to treat treatment-resistant depression. And here the study will develop and test objective methods for improving these technologies, both in terms of anatomical placement, but also using these bidirectional electrodes to obtain information from the patients to develop biomarkers that either can be used to develop longer-term strategies or even in real time to control the stimulation of these circuits. Here's a study from Drs. Darren Doherty and Alik Wij, which is using deep brain stimulation to in the internal capsule and observing how it can improve cognitive control. And they found that DBS causes an increase in theta oscillation power within the prefrontal cortex, and this is associated with improved performance in these cognitive tasks. And the change in theta oscillation is correlated with clinical outcomes in both MDD and OCD. So their current studies are focused on using these bidirectional electrodes to acquire biomarkers and longitudinal studies, which can be useful in terms of both diagnosis as well as in future therapies, perhaps in closed loop adaptive therapies. So I think the range of possibilities is really quite large and broad and high. Between these developments of technologies, a better understanding of circuits in both health and disease, the ability to analyze data in a much more sophisticated way, I really think we do have the potential to make great inroads in human therapeutics within the next 5 or 10 years. And this is what we're looking forward to supporting through not only these transformative projects that I've described to you today, but in the ongoing work supported by the BRAIN Initiative. And I just want to finish by thanking all the great folks at the NIH who've been working on this project, over 100 staff members overseeing the science, overseeing the coordination, the review, outreach, everything. It couldn't be done without their efforts. And I will stop here if there's time for questions.